You all know that life expectancy has more than doubled in the last 100 years. We have now reached 82 years in the European Union, and many diseases that were not really, diseases that were deadly a few decades ago are not really a big deal anymore. But there is one cluster of health issues that does not show this trend, and this cluster is disorders of the brain. So 25 years ago, approximately 9% of all invalidity pensions were paid due to anxiety or depression. Today, this share has increased to 43%. And what is even more alarming is that the number of teenagers who show symptoms of depression has more than doubled in the last 10 years. So there is an increasing number of disorders of the brain, also because people are becoming older. So we have a big problem, for example, with dementia, but also depression plays a big role, depression and anxiety. So now the question is, how can we solve this issue? And in the next 20 minutes or 25 minutes, I want to talk about how to use neurotechnology to treat these different disorders of the brain. So in the first part, I'm going to talk mainly about brain-computer interfaces and how they were used to restore movements and paralysis, and what we have learned from using brain-computer interfaces in a clinical environment, and how this knowledge can be translated into applications in psychiatry. So we have all now reached the age of neurotechnology, and there were a lot of science fiction movies that were already anticipating what will be possible maybe in the future. And there was also a lot of media attention in the last 5 years, I would say, because the big global companies are now also moving into the field of neurotechnology. So neurotechnology is any form of artificial means to record, analyze, and also modulate or change brain activity. And when we use neurotechnology, we can use this technology to understand, diagnose, and treat different diseases. But now, if you analyze brain activity in real time and translate it into a control signal of a digital device, you can have a new set of applications. So we can use neurotechnology to control external devices, for example, a robot or a drone. And the first, these brain-computer interfaces, they can use different forms of brain activity. What you all know is probably non-invasive means, for example, MEG or EEG, electroencephalography. Now we have seen in the last couple of years that also quantum sensors are used. These are sensors that can detect neuromagnetic activity at room temperature. So before we used squid MEG, where we had to cool down the sensors with helium so that they were superconducting to be sensitive enough to measure the activity of the brain. Today, we can do this with optically pumped magnetometers. And there is also a technique called functional near-infrared spectroscopy, where we use essentially red light to see changes in oxygenization of superficial layers of the cortex. And with this, we can see the consumption of oxygen in the brain. You all know functional fMRI, like functional MRI, magnetic resonance imaging. With this technique, we can look deeper into the brain. And when we record this activity in real time and translate it, we can also do neurofeedback with deep areas of the brain. And then obviously we can remove the skull and place electrodes into the skull, for example, superficially as an ECOG, but we can also implant. And You've probably all seen the Neuralink device that inserts threads into the brain, but the only certified device for human use is the Utah array that you see on the lower right. So here you use 100 microelectrodes to record activity of single cells or multi-unit activity. And of course, you can also use electrocorticography, as I mentioned. Yeah? So the first clinically meaningful BCI was introduced in the late 90s in a patient who suffered from locked-in syndrome, who was unable to speak or move and who depended on artificial respiration. This patient could select letters on the screen by modulating his electric activity and write a letter. So in parallel, also implantable devices were developed that record brain activity for high-dimensional control of robotic arms. Here you see a number of examples. These are all using the Utah arrays, and on the right you see a person who is using 3 Utah arrays implanted in his brain to control 2 prosthetic devices. The big problem with this is that all implantation involves the risk of infections and bleedings. There is no certification for long-term use. So usually these devices have to be removed after a while. And these systems have never been used outside of the lab in the everyday life of these patients. So there is always an engineer close by having a red button to interrupt any malfunction of the systems. Obviously, there are also a number of other disadvantages when you implant. So, for example, once you have implanted the device, the user cannot remove it at will. And also implanting these devices in hundreds of thousands or even millions of people raises a number of very complex ethical questions. So in my group, we have worked on a non-invasive BCI that we used in patients with quadriplegia. So these patients were unable to grasp these different objects of daily living, for example, the cup, a pen, or a piece of paper. And you can see they can move the shoulder and they can reach the objects, but they cannot grasp. We have equipped these patients with a neural exoskeleton. So these, the system translated their electric brain activity into actual grasping motions when they were when they had the intention to move their fingers. The system could be also used outside of the lab. So this man could go into a restaurant and order some food and eat it with a fork for the first time after many years. So we have also asked the patients what they would like to do with this ability. And here you see a patient who asked for a plate of potato chips. And you can see that the system really improved the quality of life of this man. We have also used the system in patients with chronic stroke. And these patients, they could not move their fingers for years or sometimes even for decades. And what we've seen after 1 month of daily training, when they used it for an hour on a daily basis, we've seen that after 1 month, the patients could move, some of these patients could move the fingers again. And using neuroimaging techniques, we found that their brains have reorganized and also rewired. And as you can see, before the training with the exoskeleton, their brain was equally activated on both hemispheres. After the training, we could see that the activity has shifted to the areas that were connected to the exoskeleton. So using this exoskeleton shifted their brain activity and also led to a rewiring of their brain that allowed them to use their hand even without the exoskeleton. So another example of the neuroplasticity that can be induced by neurotechnology is shown here. So this is a rat in which the spinal cord was actually cut through. So the rat should not be able to move. But using a brain-computer interface that was connecting the cortex, you see here the wire, and the spinal cord, the rat could walk again. The same principle was also implemented in a patient with a lesion you see here, and they implanted an electrode underneath the lesion that was triggered by movements of the body. So whenever the patient was leaning forward, the stimulation went on, and the patient could actually do locomotion movements. When the stimulation was turned off, the patient could not move. So here you see that the patient could move with the system. And as with the patients who used the exoskeleton, the patients used it for several months. And here you see that after these months, the patients could move their legs again. And this patient, after a few months of training, could even walk unassisted. And he was in a wheelchair before. So that is a very good example of how this kind of real-time brain-computer interfacing can induce plasticity, triggering neural recovery after stroke or after quadriplegia because of a spinal cord injury. So, well, we have now put this to a new level. So we want that the stroke patients can use the system at home, obviously, where they can also use the system for assistance, for example, in the kitchen. So we are working on systems that the patients can use at home. And that's a technical challenge. So we want that the users can use the system independently so that they can actually mount it unassisted. That's a larger project that we are doing now. And I actually wanted to talk about psychiatry. So that was just the preludium to show you what is possible with brain-computer interfaces in the motor domain. And I think it's very important to see these results because you get an idea of the potential of this technology. So in this first part, I wanted to show you that these brain-machine interfaces are effective tools to improve living conditions in paralysis, and that using these systems on a daily basis can lead to neuroplasticity and rewiring of the brain, so functional and structural plasticity. And the same principle may also work in the treatment of other disorders of the brain, for example, depression or anxiety. The question is now, how? And I want to talk about this now for the next 10 minutes. So in the motor BCI, we are translating brain activity into sensory feedback, and we translate usually the intention or the thought of a movement into an actual movement of an exoskeleton, or you can also use electric stimulation, and there are different ways to do that. Now, we have seen that this leads to functional and structural reorganization of the brain. In the future, when we want to use these systems for treatments in psychiatry, we will also translate brain activity in real time and provide some form of sensory feedback. So this sensory feedback can be very diverse. So it can be vibrotactile, vibroacoustic, it can be visual. There is no limit as long as the brain can sense the feedback. However, we want to translate brain activity related to a number of brain functions that are affected by psychiatric disorders. For example, working memory, emotion regulation, or also sensory motor integration. And we want to use adaptive brain stimulation to foster this neuroplasticity and improve brain function. Now, a logical step to identify these neural substrates, the oscillatory correlates of these different brain functions like working memory and so on, is to record brain activity, use brain stimulation, and see how this stimulation changes the brain activity and behavior. With this kind of approach, we can infer the causal links between brain activity and behavior or brain function. There is another dimension to it. So this is a very simplified concept here, and it's based on a waveform of a signal. But we know that the brain, of course, is super complex, and we have a number of neural circuits in the brain that are linked to certain brain functions. So when we want to target these different brain activity patterns, we also need to be spatially very specific. And that is a big challenge right now. So based on the work that has been done in neuroimaging, we know now that there are certain brain areas, for example, related to dysphoria. You see here these different areas that are interacting, the amygdala, the insula, also the orbital frontal cortex, the hippocampus. And you see that there is a hyperconnectivity in this network, for example, in this depression. While there is a hypoactivity between the dorsolateral prefrontal cortex and the amygdala, for example. Yeah? And we know this now already from neuroimaging studies. Now we have to find ways to be spatially specific, record activity from these very areas, and then drive them into different states to change the activity in these neural circuits. Yeah? So we know now, for example, there is a circuit for anhedonia, rumination, dysphoria, or disrupted cognitive control. So disrupted cognitive control is also very important, for example, for OCD, for obsessive compulsive disorder, and also in other disorders. So how can we record brain activity while we stimulate the brain? Because we want to know, if we know these areas, what is really the activity, what is the frequency, and what is the oscillatory waveform responsible for the communication of this area with other areas in the brain. To do this, we need to combine brain stimulation with neuroimaging. And if you do electric stimulation to the brain, it is very difficult to record activity from the brain because you see a lot of artifacts. We have developed a number of techniques to stimulate the brain with different waveforms. This is DC stimulation, while we reconstruct the activity, the physiological activity related to finger tapping, for example. Here you see an example of using a new stimulation technique called amplitude modulated alternating current stimulation to target different frequencies in the brain and reconstruct this physiological activity while the brain is stimulated. Yeah? So that was something that we established 4 years ago. And we have now also a possibility to do, this was in MEG, we have now also the possibility to do this in EEG in real time. That's very important. And it's now in the last round in NeuroImage, so it's already available as a reprint. I can only recommend to have a look if you want to do this kind of research, real-time analysis of brain activity during brain stimulation, targeting different oscillations. This work was now published by David Haslacher, who is a PhD student in my lab. So you'll see it soon in NeuroImage. So that's all possible now. We have the tools, and now we have to go and target different brain functions. So just to see whether it's possible to apply this technology, we have established this in a working memory task. So when you do a working memory task, you see an increase of theta oscillations. These are oscillations between 4 and 9 hertz in the frontal lobe. This is called the frontal midline theta. And we have targeted this oscillation, and we could see that with this, you can actually modulate the performance of working memory. And of course, you can also modulate then the power of this frontal midline theta when you stimulate at this frequency. So this is all possible. We can reconstruct that during the stimulation and really look into the brain while we target these oscillations. Now, to come back to the big challenge of spatial specificity. If we use EEG, there are a number of limitations in terms of spatial specificity. So usually for our motor BCI, we use Laplace filter or filters that give you at least an idea of left or right hemispheric sources. But what we need is something that is much, much higher. Yeah, so if you want to record, for example, from the dorsolateral prefrontal cortex, we're speaking about an area that is only 1 centimeter large, and you don't want to have activity from other areas. And here we can use quantum sensors. These are the sensors that I already mentioned, the OPMs, optically pumped magnetometers. And using these sensors that are much, much closer to the skull compared to the conventional MEG, where you have approximately 7 centimeter distance from the brain. Here you have approximately 3 centimeters distance from the brain. You reach a much higher spatial resolution. And as there's no volume conduction, you can reconstruct the sources really at a much, much higher spatial resolution. And what we also need is brain stimulation techniques that are spatially specific. And that's the next problem. So when we stimulate the brain electrically, the electric fields are finding their way through the tissue that has the lowest impedance, obviously. So the electricity goes through the eyes and through the veins. And it is very difficult to model this. So we can attempt it, but as the brain is an organ, it can change the impedance depending on how much you drink and on the food that you eat. So there are, and your physiological state. So we need a different technique, and we need something that is much more spatially specific. So this could be done with magnetic stimulation. And that is something that, of course, we are also working on, magnetic stimulation of the brain. So now, how can this new system that targets this activity in the brain be embedded in psychiatry? So first of all, we need to identify these individual neurosubstrates of brain functions. And we can then embed this in the established methodologies like psychopharmacology and especially psychosocial interventions. So context is very important. And we can also use digital tools to track the effect of the stimulation and neurofeedback. For example, a smartwatch or certain questionnaires that you use on the smartphone. That is a big gap in knowledge when I do my treatments here at the Charité. I see the patients probably once a week during the treatment, and they can, of course, tell me how the situation was. But using digital tools, I can get a much better idea of what the impact was of the stimulation. And using these digital tools, we can also record, of course, a mobile EEG. We can objectify what has changed in the brain and create computational models that help us to even improve the whole circuit of BCI neurofeedback and stimulation. So let's come to the final conclusions. I have shown that repeated use of a brain-computer interface can result in functional and structural neuroplasticity. Using brain stimulation at the same time can elucidate the causal link between brain activity and brain functions and behavior, and that not only in the motor domain, but beyond that, working memory, emotion control, cognitive control, all these different brain functions. Now, what is key is really phase frequency and spatially specific targeting of this brain activity. Yeah. So for this, we probably need more advanced techniques than we have right now, like EEG or electric stimulation of the brain. We can do something with it, but we need to refine these techniques. So quantum sensors that I introduced and novel brain simulation techniques are very promising tools to advance this tool set of neurotechnology towards applications in psychiatry. And with this, I would like to stop here, and I'm very happy to answer some questions now.